Assalamu alaikum. In the chapter of cell, the unit of life, today we are going to discuss about another example of another mechanism of active transport, that is endocytosis and exocytosis. So firstly we must know about what is endocytosis. Endocytosis term was coined by Christian D. Tube in 1963. So what is endocytosis? According to definition, endocytosis is the process by which cell takes in the substances from outside of the cell by engulfing them in a vesicle. Okay, so in the process of endocytosis, cell takes in some substance in the form of vesicle. Means in the process of endocytosis, firstly the vesicle is formed and then the substance is taken in. So endocytosis is of two types. Number one is the phagocytosis, number two is the pinocytosis. In case of phagocytosis, some solid thing is involved, means something which is solid is taken in, whereas in case of pinocytosis, something which is liquidy or water in nature is taken in. So this diagram shows the difference between phagocytosis and pinocytosis. So this is the extracellular environment, and here this is the inside of cell. So this is inside of cell, or you can say this is cytosol or cytoplasm. So according to the definition, firstly, in case of endocytosis, firstly vesicle is formed. As you can see here, this is the vesicle. The plasma membrane invaginates, means where plasma membrane enfolds inside, or plasma membrane shows the enfolding towards inside, and vesicle is formed. So firstly there is a vesicle, and in case of phagocytosis, this is solid particle. This solid particle goes inside the vesicle, and after that in the next step this vesicle is pinched in or is taken in, and now you can see here the food particle along with the small piece of plasma membrane is present inside the cell. So now this is called the phagosome or food vacuole, because this is the vesicle, or inside that the food is present, that's why it is called the food vacuole. Whereas in case of pinocytosis or pinocytosis, the process is same. Firstly, the vesicle is formed by the invagination of plasma membrane, or plasma membrane shows the enfolding towards the inside, and after that this is the watery substance or extracellular fluid. This extracellular fluid goes inside the vesicle, and after that the vesicle is pinched off, and in the next step this vesicle is taken in, and now this vesicle contains the extracellular fluid. So there is a difference between phagocytosis and pinocytosis. In case of phagocytosis, solid particle is involved, whereas in case of pinocytosis, the extracellular fluid is involved. Otherwise the process is same. In the first step, the vesicle is formed, and after the vesicle, how vesicle is formed? Vesicle is formed by the enfolding of plasma membrane, and after that the particle or solid particle or extracellular fluid is taken in, and in the third step the vesicle pinched off, and in the next step the vesicle is inside the cytoplasm. Now this is the detailed process of the pinocytosis. As you can see, this is the plasma membrane, and here the solute particles are present. So firstly the plasma membrane shows the enfolding, and the vesicle is formed, or you can say vesicle is a cup like structure. So this vesicle is formed, and in the next step the extracellular fluid goes inside the vesicle, and in the next step plasma membrane continues to fold and it forms a closed cup shaped structure, and in the last step the vesicle is taken in having the extracellular fluid. Now this structure is called the pinosome. Some means body, and pino means watery substance, means this body contains the watery substance. So this is the process for the pinocytosis. Means firstly the vesicle is formed. In the second step the extracellular fluid goes inside the vesicle, and then the vesicle is closed, and after that the vesicle is taken in. And in case of pinocytosis, the vesicle is called the pinosome, that is having the extracellular fluid. Whereas this diagram shows the process of phagocytosis. As you can see, firstly, in the case of phagocytosis, there may be anything, like it may be food particle, or maybe the microbes, or any other thing. So whether it is a food particle or microbe, they both are in solid form. So firstly the vesicle is formed, and after that the solid material, either in the form of food particle or microbe, is taken in, and this is the phagosome. And after the formation of phagosome, here in the cell the lysosome is present. Lysosome contains the hydrolytic enzyme for digestion. So this lysosome attaches to the phagosome, and here between the phagosome, and as you can see, this is the phagosome, here the lysosome comes and attached to the phagosome. As you can see here, the wall is present for the lysosome and the wall is present for the phagosome. In the next time the wall dissolves, and after the dissolution of wall, this hydrolytic enzyme starts to hydrolyze the material, or start to digest the material. So after the digestion, the material is absorbed by the cell, the useful material is absorbed by the cell. So this is the useful material which is absorbed by the cell, and if the material is not useful, then this material is dumped outside the cell. So this is the whole process of phagocytosis. Means in the first step the material is taken in, and after that the lysosome comes and binds to the phagosome, and after that wall dissolution takes place, and then the material is digested by the hydrolytic enzymes of lysosome, and the materials are absorbed, and the residual body, or non-essential material, is dumped outside the body. So next definition is exocytosis, because if something is taken in, then it is called the endocytosis. Means something is taken in inside the cell, but here there is a terminology that is exo, means outside. Means after that, if the material is inside the cell, if the material is taken in and the material is absorbed by the cell, so if there is some non-necessary material, or there is some residual material is present, then there is a need to throw out the residual material. So in that case the exocytosis process works. So what is the definition of exocytosis? Exocytosis is the process of moving material from within a cell to exterior of the cell. Means pumping out from inside to outside. As you can say here, here you can say this is the residual material. Means this material is not residual material. As you can see, this material is not needed by this cell. So in that case, for the process of exocytosis, this is the reverse of phagocytosis, or you can say endocytosis. Firstly the vesicle is formed, the vesicle attaches to the plasma membrane. So this vesicle attaches to the plasma membrane, and after that here the wall, or this opening, or this pore opens up, and after that this material is dumped out of the cell. So this is the just the reverse of endocytosis. In case of endocytosis, firstly the vesicle is formed and the vesicle is pinched in, and now the vesicle is inside the cell. Whereas in case of exocytosis, firstly vesicle attaches to the plasma membrane, the pore opens up, and now the material is thrown outside the cell. And now because in the process of endocytosis, the vesicle is formed from the part of plasma membrane. As you can see, this vesicle is formed from the part of plasma membrane. So in the case of exocytosis, this part of plasma membrane is given back. So in that case, the surface area of the plasma membrane is maintained. If it is used in the case of endocytosis, then the same piece of plasma membrane is given back to the plasma membrane during the process of exocytosis. So in that manner the surface area of the plasma membrane is maintained. If it were not happened, then in that case every time if small piece of plasma membrane is taken in and is not given out, or then in that case every time the size of the or the surface area of plasma membrane is reduced. So this process is very important. Exocytosis process is very important to maintain the surface area of plasma membrane.